The overall goal of this procedure is to accurately determine the relative levels of post-translational modifications within preparations of total cellular nucleosomes obtained from mammalian cells. This is accomplished by first preparing high-quality homogeneous cultures of mammalian cells. Then, nuclei are isolated from the cells through mechanical disruption with a Downs homogenizer and centrifugation. The next step is to obtain intact nucleosomes by digesting the chromatin present within the nuclei and performing a series of hypotonic extractions. Lastly, the nucleosomes are interrogated using an ELISA assay with the appropriate controls to identify specific post-translational modifications. Ultimately, results can be obtained that show the relative levels of specific modifications present within nucleosome samples.
The main advantage of this technique over existing methods like Western blotting and chromatin immunoprecipitation is that a global picture of a number of nucleosome modification states can be readily determined. The method is much more sensitive and accurate than Western blotting and can be performed in most labs equipped for general molecular biology experimentation.
This method can help answer key questions in the stem cell and epigenetics field, such as what happens when major differentiation and reprogramming events occur.
To begin the procedure, first trypsinize the cells with 3 ml of trypsin per 15 cm plate. To freshly trypsinized cells, add 20 ml of ice-cold PBS butyrate, transfer the cells to a 50 ml conical tube, and centrifuge the tube to collect the cells at 1000 rpm for 5 minutes. Aspirate the supernatant and add 10 ml of ice-cold PBS butyrate to wash. Centrifuge the cell suspension again at 1000 rpm for 5 minutes. Remove the supernatant and resuspend the cell pellet in 4 ml of lysis buffer with protease inhibitors, as described in the written protocol. Next, pipette the cells to a type B pestle Downs homogenizer on ice. Downs homogenize with 20 strokes. Then, transfer the homogenate to a centrifuge tube on ice. Centrifuge the sample at 2000 G for 10 minutes at 4 degrees Celsius. Remove the supernatant, which contains the cytoplasmic material, and resuspend the pellet in 2 ml of ice-cold wash buffer C with protease inhibitors. Gently layer the resuspended material onto a 5 ml 30% sucrose cushion in a cold centrifuge tube. To isolate the nuclei, centrifuge at 2400 G for 5 minutes in a swing bucket rotor. Nuclei will migrate through the cushion and cellular debris remains at the interface. After centrifugation, carefully remove all liquid volume and resuspend the nuclei in 250 microliters of ice-cold wash buffer C with protease inhibitors. Transfer the nuclei to a 1.5 ml microcentrifuge tube. To begin isolation of nucleosomes, add 3 microliters of 0.1 molar calcium chloride to the nuclei and place in a 37 degrees Celsius heat block until the temperature is equilibrated. Then, add 2 units of micrococcal nuclease in 10 microliters of micrococcal nuclease buffer and incubate for 12 minutes at 37 degrees Celsius. Mix frequently with a pipette tip. After the incubation, stop the reaction with 6 microliters of 0.5 molar sodium EDTA pH 8 and place on ice to cool. Next, centrifuge the sample at 2000 G for 4 minutes. Following centrifugation, discard the supernatant and resuspend the pellet in 300 microliters of 0.2 millimolar sodium EDTA. Incubate the sample on ice for 1 hour with occasional gentle pipetting to mix. Following a centrifugation at 3000 G for 4 minutes at 4 degrees Celsius, collect the supernatant, which contains the free nucleosomes, in a new microfuge tube and store on ice. To liberate additional nucleosomes, resuspend the pellet in 300 microliters of 0.2 millimolar sodium EDTA as before, and incubate on ice for 1 hour with occasional gentle mixing. After the incubation, centrifuge the sample again. Combine the resulting supernatant with the remaining sample in the microfuge tube on ice. To begin the nucleosome ELISA assay, load a 96-well ELISA plate with a descending series of 5 two-fold serial dilutions of nucleosomes in coating buffer, starting with 0.1 micrograms per 50 microliters in the top well. All dilution series should be loaded in triplicate. Remember to include wells with coating buffer only as controls. Incubate the plate overnight at 4 degrees Celsius. The next day, use a plate washer to wash the plates with 200 microliters per well of 0.5% tween 20 and PBS at room temperature 4 times for a combined total of 10 minutes. Now, add 100 microliters per well of blocking solution and incubate for 1 hour at room temperature on a rotator. After the incubation, remove the blocking buffer by shaking the inverted plate briskly over a sink. Next, add the diluted primary antibody in a volume of 50 microliters per well in a solution of 5% BSA and 0.05% tween 20 and PBS. Incubate the solution for 1 hour on a rotator at room temperature. After the primary antibody incubation, wash the plates as before using the plate washer. Once the wash procedure has completed, add 50 microliters per well of horseradish peroxidase conjugated secondary antibody diluted in PBS with 5% BSA and 0.5% tween 20. Incubate at room temperature for 1 hour on a rotator. Following secondary antibody incubation, wash the plates as before using the plate washer. To develop the plate, add 50 microliters of TMB ELISA substrate to each well and incubate for 10 minutes at room temperature. The wells of the ELISA plate will turn blue and the intensity is proportional to the amount of nucleosome modifications present within each well. Then, stop the reaction by adding 50 microliters per well of 2-normal sulfuric acid. The color will change to brown. Centrifuge the plate at 1500 RPM for 2 minutes to dissipate any air bubbles. The plate is now ready for quantification on a colorimetric plate reader. Finally, read the plate at 450 nanometers and export the results for analysis.
Once mastered, this technique can be done in 2 days if it is performed properly.
After its development, this technique opened the door for researchers in the field of stem cell research to explore epigenetic responses to differentiation and reprogramming events at the level of the entire epiproteome.